Hi, am I audible clearly? If you can confirm once. Uh, Hello. Okay. I think my voice is clear now. So I'll start the session. So today's seventh problem solving session for the course overview and integration of cellular metabolism. The course instructors are Professor Aditri Veer and Professor Arindam Ghosh from IIT Kharagpur, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. So, till now we have discussed about carbohydrate metabolism and lipid metabolism. Today we'll start protein metabolism. So, today we'll discuss different questions related to the protein metabolism parts that are discussed in seventh week session. Uh, I have prepared certain questions related to your assignments, this week's assignment. So, feel free to ask any questions in the middle, or if you have any doubt in any of the topics discussed in this week. Okay, I hope I can start the class now. Starting with the first question: where does protein digestion begin? Option A, mouth. Option B, stomach. Option C, small intestine. And option D, colon. Where does protein digestion begin? Mouth, stomach, small intestine, or colon? The correct answer to this question is option B, stomach. So, we don't have any enzymes to digest the protein in the saliva. So, there's almost no digestion of protein in the mouth. So, the digestion of proteins, whatever we are taking in our diet, that begins in the stomach. Okay. So, once we whatever dietary proteins are there, whatever we are eating, they have multiple sources of protein, right? In those sources, so the digestion, it does not start in the mouth, it starts from the stomach. So we have different enzymes in the stomach and then pancreas and intestine which can digest the proteins. So these are called proteolytic enzymes. Proteolytic enzymes because it can lyse the protein. So lytic, the term lytic comes from lysis. Okay, and lysis means breakdown. So we are breaking down the protein. Basically, digestion means breakdown of food. So you have read about glycolysis. That was basically breakdown of carbohydrate into glucose and then breakdown of glucose into further smaller compounds. Okay. So similarly, now we are talking about protein digestion. So it starts from stomach. We have enzymes like pepsin and renin. Renin is mainly present in child, and it is used to break down the milk protein. And pepsin, again, it is present in stomach. So these two enzymes, you know that pH of stomach is acidic. So these two enzymes work well in acidic environment. Okay. Then comes enzymes in the pancreas, that are trypsin, chymotrypsin, elastase, and carboxypeptidase 1 and carboxypeptidase A and B. Then there are other enzymes present in intestine that are aminopeptidases, dipeptidases, and tripeptidases to further break down the protein. So when we eat different sources of proteinaceous food, all the protein enter the stomach first, where pepsin and renin they will digest the respective proteins, and then those digested proteins will go through the intestine, where they'll be further broken down into smaller peptides, and finally to into single amino acids, and then it will be absorbed in our body. Okay, and those amino acids will be recycled and used for making different cellular proteins that are required by our body. So that's how, like, this is a very basic gist of protein digestion. I hope this is clear. Moving on to the next question. How is pepsinogen activated? Option A, trypsin. Option B, enteropeptidase. Option C, HCl. And option D, amylase. How is pepsinogen activated? Option A, trypsin. Option B, enteropeptidase. Option C, HCl. And option D, amylase. You can write the answer in the chat box. So the correct answer to this question is option C, HCl. So pepsinogen is a zymogen. So all these proteolytic enzymes, they are mainly present in the form of zymogen. Okay, what are zymogens? Zymogens are the inactive form of the enzymes. Okay, so since these proteins are proteolytic, these are proteases, okay, or proteolytic enzymes. So since these enzymes are proteolytic enzymes, they can degrade any protein, right? So, and if they are always activated, they will start chopping off your cellular proteins as well. That's why there's a need to make them inactive when not required by our body. So, if there's no dietary protein or any other protein that has to be degraded, if there are no such proteins present in the environment, these proteolytic enzymes remain inactivated. Only when they are required, they are converted from the inactive zymogen to the active enzyme form. Okay, from inactive zymogen form, it is converted to active enzyme form. Okay, so pepsinogen is one such zymogen. So it is a zymogen of pepsin. So pepsinogen is converted to pepsin. So in stomach, pepsin is present in the form of pepsinogen. Okay, and it is converted to pepsin by the HCl that is present in the stomach. So when we eat food, our stomach secretes the acids for the protein digestion. And first, this acid uh, converts this pepsinogen. So activates this pepsinogen into pepsin. And then this pepsin can act on the proteins to break them down. Okay. So HCl basically activates the pepsinogen. How does it do? Like, what does it do? So you know, hydrochloric acid is secreted in the stomach, as I've mentioned, and the optimum pH is 2 to 3. What uh, what HCl does is HCl basically cleaves 44 amino acid from the N terminal of the pepsinogen. So pepsinogen is the zymogen form of the protein, and from that, these 44 amino acid from the N terminal end is cleaved off by the HCl. That makes the remaining protein active, and that is called pepsin. And pepsin catalyzes the hydrolysis of the bonds formed by the carboxyl groups of phenylalanine, tyrosine, tryptophan, and methionine. Okay, so this is how pepsin breaks down the protein. So it will chop off the protein wherever the phenylalanine or tyrosine or tryptophan or methionine is present in the protein. Okay, so it converts the protein into proteoses or peptones. So it converts the proteins into smaller peptones or proteoses. Okay, and then these go into the into the intestine, where other enzymes like trypsin, chymotrypsin, they will act on these uh, smaller peptones or proteoses, and they will break them down further into amino acids. Okay. So how pepsinogen is activated? It is activated by HCl. How does HCl activate it? HCl removes 44 amino acids from the N-terminal end of the pepsinogen to make it pepsin. Once we have the active pepsin, pepsin will then catalyze the hydrolysis, of the peptide bonds formed by the carboxyl groups of phenylalanine, tyrosine, tryptophan, and methionine. Okay, so you have to remember these four amino acids where pepsin is doing its action, and you have to remember that how many amino acids are removed from the pepsinogen to make it pepsin, and what activates pepsinogen into pepsin? It is HCl. HCl, from where we are getting the HCl? It is secreted from the stomach. Okay, I hope this is clear. Moving on to the next question. Chymotrypsin cleaves peptide bonds specifically adjacent to which amino acid? Chymotrypsin cleaves the peptide bond specifically adjacent to which amino acid? Option A, phenylalanine and tyrosine. Option B, leucine and isoleucine. Option C, methionine and valine. Option D, aspartic acid and glutamic acid. And option E, arginine and lysine. Chymotrypsin cleaves the peptide bond specifically adjacent to which amino acid? Phenylalanine and tyrosine. Option B, leucine and isoleucine. Option C, methionine and valine. Option D, aspartic acid and glutamic acid. And option E, arginine and lysine. So the correct answer to this question is option A, phenylalanine and tyrosine. Okay. So chymotrypsin basically cleaves the peptide bonds adjacent to aromatic amino acids. So tryptophan, tyrosine, and phenylalanine, these are the aromatic amino acids. Aromatic amino acid means the amino acids that has a aromatic side chain. So you all know the basic structure of amino acid, which has a central carbon to which a carboxyl group is attached, amino group is attached, and a hydrogen is attached, and then we have R group. So this, depending on this R group, we have 20 different amino acids present in nature, right? So if this R group has an aromatic group, so aromatic group, to in simple words, if it has a benzene ring structure, okay, so any amino acid where this benzene ring structure is there on the R group, Those amino acids are called aromatic amino acids, and we have three such amino acids: phenylalanine, tyrosine, and tryptophan. Okay. So chymotrypsin cleaves the peptide bonds adjacent to aromatic amino acids. So phenylalanine, tyrosine, and tryptophan. So option A is correct answer. Okay. So we studied about pepsin in the previous slide. So pepsin was cleaving what all amino acids? Pepsin was cleaving phenylalanine, tyrosine, tryptophan, and methionine. So along with phenylalanine, tyrosine, tryptophan, it was also cleaving methionine. Chymotrypsin cleaves phenylalanine, tyrosine, and tryptophan. Okay, I hope that's clear. Moving on to the next question. Which of the following enzyme is not a part of digestive enzymes in the small intestine? Which of the following enzymes is not a part of digestive enzymes in small intestine? Option A, trypsin. Option B, chymotrypsin. Option C, elastase. And option D, pepsin. Which of the following enzymes is not a part of digestive enzyme in the small intestine? Option A, trypsin. Option B, chymotrypsin. Option C, elastase. And option D, pepsin. So the correct answer to this question is option D, pepsin. So pepsin is the enzyme that is present in stomach. We saw in the first question where pepsin is present. Pepsin is present in the stomach at the acidic pH. It works at the acidic pH. Whereas trypsin, chymotrypsin, and elastase, they are present in the small intestine and they work at a a near neutral pH. So the pH of small intestine is around 8. So optimum pH for trypsin, chymotrypsin, and elastase is pH 8. Whereas optimum pH of pepsin is pH 2 to pH 3. Okay. So pancreatic juice contains endopeptidases that are trypsin, chymotrypsin, and elastases. All the three of them are serine proteases. They have a serine group in their active site. And then there are exopeptidases that are carboxypeptidases. The peptide hormones, uh, cholecystokinin and pancreozymin stimulates the secretion of pancreatic juice secreted as all of these enzymes are secreted as zymogens, as I have already mentioned, and secretion of alkaline bile and pancreatic juice occurs together. So I told you that all these enzymes are active at a alkaline pH, that is pH 8. Okay. So we have studied about pepsin. Pepsin was working at acidic pH, and it can cleave the amino acids. It can cleave the protein after the amino acids phenylalanine, tryptophan, tyrosine, and methionine. We have studied about chymotrypsin. It can cleave at phenylalanine, tryptophan, and tyrosine, basically aromatic amino acids. Okay. Trypsin can cleave after arginine and lysine. Okay. And all of these I've mentioned, they are secreted as zymogen, that is the inactive form of the enzyme, and they are activated when they are required by the body. Okay, next question is trypsinogen is activated to trypsin by the enzyme. Option A, pepsin. Option B, enteropeptidase. Option C, aminopeptidase. Option D, carboxypeptidase. And option E, dipeptidase. Trypsinogen is activated by the enzyme. Option A, pepsin. Option B, enteropeptidase. option C, aminopeptidase, option D, carboxypeptidase, and option E, dipeptidase. So trypsinogen is activated to trypsin by the enzyme. Option A, pepsin. Option B, enteropeptidase. Option C, aminopeptidase. Option D, carboxypeptidase. And option E, dipeptidase. You can write the answer in the chat box if you know. The correct answer to this question is option D, enteropeptidase. So I've mentioned already that uh, these proteases, they are secreted as zymogen. That so like we saw in the previous question, we had pepsinogen as a zymogen form, and the activated form was pepsin. Similarly, trypsinogen is the inactive zymogen form, and the activated form is trypsin. So when trypsin is required by the body, trypsinogen will be activated uh, into trypsin, and this activation is catalyzed by another enzyme, which is enteropeptidase or enterokinase. So what does this enzyme do? It cleaves a hexapeptide out of the trypsinogen to make it active trypsin. Okay, so fully functional active form is made from trypsinogen by removing the hexapeptide, 6 amino acid long chain. Okay, the hexapeptide. So it is activated by enterokinase. This enterokinase is present in the intestinal microvillus membranes. So removal of hexapeptide from the N-terminal end, as I've mentioned. So if you remember from the pepsinogen activation, how many amino acids were removed from the N-terminal end? For pepsinogen activation, HCl was doing the work and it was removing 44 amino acids from the N-terminal end. Here, for trypsinogen activation, enterokinase is doing the job and it is removing 6 amino acids, basically a hexapeptide from the N-terminal end. And we have the trypsin, that is the active form, and it can do its once we have some amount of trypsin, it can it can conduct autocatalysis, where it can make more and more trypsin from trypsinogen by doing this cleavage. Okay, so it can do autocatalysis and it can activate other enzymes. Trypsin catalyzes the hydrolysis of the bonds formed by carboxyl groups of arginine and lysine. So you remember cleaves pepsin cleaves at the carboxyl end of phenylalanine, tyrosine, tryptophan, and methionine. Okay, chymotrypsin. Chymotrypsin is cleaving at aromatic amino acid, carboxyl group of aromatic amino acid. Okay, now trypsin is cleaving at the carboxyl group of arginine and lysine. Okay, so you have to remember the sites where these proteases act. Okay, so we have pepsin, we have chymotrypsin, and we have trypsin. So there's a condition called acute pancreatitis where uh, premature activation of trypsinogen inside the pancreas itself will result in the autodigestion of pancreatic cells. So basically, I told you that why these zymogens are active, why these zymogens are important. These zymogens are the inactive form of the uh, proteolytic enzyme. Now, if these zymogens get activated at a premature stage when it is not required, what it will do, since these are proteolytic enzymes, they are going to chop off any and every protein that comes in contact with these proteolytic enzymes. Okay, so if trypsinogen is activated at an early stage when it is not required, when there are when there are no proteins present, what they will do, since they are activated, now we have active trypsin, it will start digesting the cellular protein. So it will start digesting the pancreatic cells, the proteins present in the pancreatic cells, and uh, slowly the cells will be degraded. So this will create a kind of autoimmune responses, because our own body will start eating its own cells. Okay, so that's it. It's an acute condition. It's called acute pancreatitis, because the pancreatic cells are auto digested by the prematurely activated trypsinogen. Okay, I hope this is clear. Moving on to the next question. Which of the following enzyme activates chymotrypsin? So now we studied about pepsin, we studied about trypsin activation. Now we will study about chymotrypsin activation. So like pepsin and trypsin, chymotrypsin is also present in a zymogen form called chymotrypsinogen. So which enzyme activates chymotrypsinogen into chymotrypsin? Option A trypsin, option B amylase, option C elastase, and option D pepsin. Option A trypsin, option B amylase, option C elastase, and option D pepsin. Which of the following enzymes activate chymotrypsin or activates chymotrypsinogen? Option A trypsin, option B amylase, option C elastase, and option D pepsin. The correct answer is option A trypsin. So I told you in the previous slide only that trypsin, once it is activated from trypsinogen, the active trypsin can do autocatalysis, and it can activate other enzymes. So chymotrypsin is one of the other enzymes. So trypsin can go and act. Once trypsin is activated, trypsin can further activate chymotrypsin. They both, so you remember trypsin, chymotrypsin, and elastase, they are present in the intestine, right? In the pancreatic juice. So uh, first trypsin is activated by enterokinase, which are present in the pancreatic microvilli membrane. And then once trypsin is activated, trypsin will go and activate chymotrypsin. So that's how all these enzymes get activated, and then they start chopping off the proteins that are present. Okay. So how chymotrypsin is activated? Uh, so chymotrypsin actually is present in the form of chymotrypsinogen, that is the zymogen form. And there are three segments. If you see here, there are three segments. So this is first segment, this is the second segment, and this is the third segment. So if you see the blue part, so there are three segments, 1, 2, and 3. And the yellow lines show the disulfide bond. Okay, yellow lines are showing disulfide bonds. Okay, disulfide bonds are basically the bonds between the sulfur-sulfur groups, so sulfhydryl groups. Uh, and these disulfide bonds are actually holding these segments together. So what happens when the hydrolysis begins? First, uh, hydrolysis is after the arginine residue at the 15th site. So here first break is made by trypsin. So I told you trypsin cleaves after arginine and lysine, right? So there is a arginine residue present here. So trypsin will go and cleave this first. So we'll get two different segments, but these two different segments are also connected by the disulfide bond. They are not separate, okay? They just become two different subunits which are connected by disulfide bond, and it is called pi chymotrypsin. Okay, so from chymotrypsinogen, we get pi chymotrypsin. This is then self digested by self digested at the leucine, tyrosine, and asparagine residue. Okay, to form alpha chymotrypsin, that is the active enzyme. Okay, so and then two different dipeptides are released. So one cleavage is made here and two cleavages are made here. Okay, so leucine residue, tyrosine residue here, and asparagine residue here. Okay, and two uh, dipeptides are released from the segment, and we have alpha chymotrypsin, which is the active form of the enzyme, which has three subunits which are connected by disulfide bonds. So it's not three different proteins altogether. They are subunits. They are connected by disulfide bond. So chymotrypsin consists of three different subunits, which are initially in the inactive form part of one whole subunit, and then they are cleaved to form three different subunits. Okay. So these three segments are approximated so that the active sites are formed. Hydrolysis, it hydrolyzes the peptide bond formed by the carboxyl group of aromatic amino acids. I have already told you. Chymotrypsin cleaves, where does it cleave? It can cleave at phenylalanine, tyrosine, and tryptophan. Okay, the aromatic amino acid uh, containing aromatic group containing amino acids. Okay. This is clear how chymotrypsinogen is converted into chymotrypsin. The active form is chymotrypsin, the inactive form is chymotrypsinogen. First, chymotrypsinogen is cleaved by trypsin, and then self digestion occurs where two dipeptides are released and three different segments which are connected by disulfide bond forms, which is the alpha chymotrypsin or the active form of chymotrypsin. Okay? And it can, so this active form, this alpha form, can then go and cleave the proteins at the aromatic amino acid residue. Okay. I hope that's clear. Moving on to the next question now. Which statement best describes the function of Meister cycle or gamma glutamyl cycle in amino acid metabolism? Which statement best describes the function of Meister cycle or gamma glutamyl cycle in amino acid metabolism? Option A, it synthesizes essential amino acids in the liver. Option B, it enables translocation of amino acids across cell membranes via glutathione dependent transport. Option C, it degrades amino acids into ammonia for urea cycle entry. And option D, it produces ATP by oxidizing amino acid directly. Which statement best describes the function of Meister cycle or gamma glutamyl cycle in amino acid metabolism? Option A, it synthesizes the essential amino acid in the liver. Option B, it enables translocation of amino acids across cell membranes via glutathione dependent transport. Option C, it degrades amino acid into ammonia for urea cycle entry. And option D, it produces ATP by oxidizing the amino acid directly. What would be the correct answer? The correct answer is option B. It enables translocation of amino acids across cell membranes via glutathione dependent transport. Okay, so Meister cycle is the cycle where it uses glutathione to transport different amino acid across the cell membrane. So all the amino acids, they cannot cross the cell membrane. So glutathione acts as a transporter where it carries the amino acid as a glutamyl amino acid, and then it translocates it across the membrane. Okay. What is glutathione? Glutathione is actually a tripeptide made from glutamate, 16, and glycine. Okay. So it is glutamate, cysteine, and glycine. It's a tripeptide. So it's called gamma glutamyl cystinyl glycine. So you should remember glutathione is also called gamma glutamyl cystinyl glycine. It's a tripeptide made of glutamate, cysteine, and glycine. And what it does, it can translocate amino acids from outside the membrane to inside the membrane. It can basically act as a transporter. Okay, so you can see the amino acid outside the membrane. They cannot pass the membrane directly. So what they do, they get attached with this glutathione to form glutamyl amino acid, and cysteine and glycine are released. This cysteine and glycine goes for the recycle of glutathione. Glutamyl amino acid will release the amino acid inside the cell. So amino acid from outside the cell is taken up by the glutathione to make glutamyl amino acid. And this glutamyl amino acid will then deliver the amino acid inside the cell and get converted into oxoproline. This oxoproline, by the action of oxoprolinase, get converted into glutamate. This glutamate, along with cysteine and glycine, will form glutathione. So first cysteine will get attached to make glutamyl cysteine by the enzyme gamma glutamyl cysteine synthetase, and then we will have glutathione synthetase where one glycine will add, and we'll get gamma glutamyl cystinyl glycine, which is the tripeptide also known as glutathione. Okay, then again the cycle can go on. So amino acid enters the cell membrane with the help of glutathione by the enzyme gamma glutamyl transferase. It will take the amino acid along with glutathione and make glutamyl amino acid. So whatever amino acid is there, just for example, we have lysine here. So lysine, so we'll get here glutamyl lysine, and cystenyl glycine will be released. Cystenyl glycine can then break down into cysteine and glycine, and then that can go for the recycling of glutathione. This glutamyl amino acid will release the amino acid and get converted into oxoproline by the enzyme gamma glutamyl cyclotransferase. This oxoproline will then get converted into glutamate by oxoprolinase, which will require one ATP hydrolysis. Then glutamate will get attached to cysteine first to make glutamyl cysteine by the enzyme gamma glutamyl cysteine synthetase. Now, this is synthetase. I've told you in the previous classes also, if it is a synthetase, it will require ATP. If it is synthase, it won't require ATP. So now it is a synthetase, so it will require one ATP hydrolysis. Glutamyl cysteine will then become glutathione by addition of glycine to it, and the enzyme is glutathione synthetase. Again, synthetase is there, so one ATP hydrolysis is occurring. This glutathione can again cycle go into another cycle. Okay, so this cycle is called Meister cycle or gamma glutamyl cycle. In intestines, kidney tubules and brain, the cycle occurs. Absorption of neutral amino acid can occur through this pathway. Tripeptide glutathione, I've told you, gamma glutamyl cystinyl glycine reacts with the amino acid to form gamma glutamyl amino acid that is then cleaved to give free amino acid inside the cell, and the net result is the transfer of an amino acid across the membrane, catalyzed, and it is catalyzed by gamma glutamyl transferase, which I have already told you. Okay. So I hope this is clear how Meister cycle is employed to transport or translocate amino acid from outside the membrane to inside the membrane or inside the cell. Okay. Clear to everyone? Moving on to the next question now. Transamination is the process of transferring an amino group from an amino acid to. Transamination is the process of transferring an amino group from an amino acid to. Option A ketone body, option B, a fatty acid, option C, a nucleotide, and option D, an alpha keto acid. Transamination is the process of transferring an amino group from an amino acid to option A ketone body, option B a fatty acid, option C a nucleotide, and option D an alpha keto acid. The correct answer to this question is option D, an alpha keto acid. So transamination, by the name itself, you can understand transamination means transfer of amine group from one amino acid to another. And how does it happen? It happens with the help of an alpha keto acid. So the amine group is basically transferred to the alpha keto acid to make a different amino acid. So you can see here, we have one amino acid. The amine group will be transferred to this alpha keto acid at this position, and we'll get a different amino acid and a different alpha keto acid. Okay, so the R1 group that was there in this amino acid will now be present in the alpha keto acid, and the R2 group that was present in the keto acid will be now present in the new amino acid that is forming. Okay, one example is transfer of amine group from aspartate to form glutamate. So these reactions are actually, this transamination is mostly done for amino acids to form glutamate finally, which can then be processed by deamination. So aspartate will transfer its amino group to oxaloacetate. Sorry, aspartate will transfer its amino group to alpha ketoglutarate to form oxaloacetate. So how does the reaction go? What the reaction will do? So this enzyme, aminotransferase or transaminase, we have the enzyme that catalyzes the transamination. It is called aminotransferase since amino group is transferred, or it is also called transaminase. Okay, it is also called transaminase. So what does transaminase do? It will transfer the amine group from aspartate. So this enzyme has a cofactor PLP. What is PLP? PLP is pyridoxal phosphate. Okay, this is a cofactor present. This is the cofactor of the enzyme aminotransferase or transaminase. This cofactor will first take the amino group, become pyridoxamine phosphate, that is PMP. So pyridoxal phosphate, from pyridoxal phosphate, the aldehyde group will take the amine group and become pyridoxamine phosphate, and then transfer this and then in return we'll get oxaloacetate as the product. If we remove the amine group from the aspartate, we'll get oxaloacetate, right? And this amine group that is carried by pyridoxamine phosphate, it will donate this amine group to alpha ketoglutarate, and then alpha ketoglutarate will become glutamate. Okay, so it is all this whole mechanism is called ping pong mechanism. because one in substrate is converting into a product, and then second substrate is converting into a product. So we have two substrate, aspartate and alpha ketoglutarate. First aspartate is getting converted into oxaloacetate, and then alpha ketoglutarate is getting converted into glutamate. Okay, so it's called a ping pong mechanism. Okay, so the enzyme is transaminase or aminotransferase, or both the names can be used interchangeably. The cofactor is pyridoxal phosphate, pyridoxal phosphate. It carries the amine group to become pyridoxamine phosphate and transfers the amine group to the second substrate to again get recycled inside the enzyme. Aspartate transfers its amine group to alpha ketoglutarate indirectly by the pyridoxal phosphate mechanism, and aspartate becomes oxaloacetate and alpha ketoglutarate becomes glutamate. As a result, we get from one amino acid and one alpha keto acid, we get a different amino acid and a different alpha keto acid. Okay, I hope this is clear. So transamination is the process of transferring an amino group from an amino acid to an alpha keto acid to make a different amino acid and a different alpha keto acid. Moving to the next question now. The enzyme responsible for the conversion of ammonia into urea is option A, alanine transaminase, option B, glutamate dehydrogenase, option C, carbamoyl phosphate synthetase 1, CPS1, and option D, glutamine synthetase. The enzyme responsible for the conversion of ammonia into urea is option A, alanine transaminase, option B, glutamate dehydrogenase, option C, carbamoyl phosphate synthetase 1, CPS1, and option D, glutamine synthetase. Alanine transaminase, glutamate dehydrogenase, carbamoyl phosphate synthetase, CPS1, and glutamine synthetase. The correct answer is option C, carbamoyl phosphate synthetase 1 or CPS1. So this is the first step in the formation of urea. So from ammonia, uh, since we are since we excrete the nitrogen as urea, from ammonia it gets converted into urea, and the first enzyme that is involved in this whole pathway is carbamoyl phosphate synthetase 1. What it does, it basically converts the ammonia into carbamoyl phosphate. So carbamoyl phosphate synthetase, since again synthetase is there, it will require ATP. And if you see, the reaction is between the ammonia and the bicarbonate. So ammonia and bicarbonate, or ammonium ion and bicarbonate ion, will react and form carbamoyl phosphate. Okay. So phosphate group is coming from ATP. So two molecules of ATP are required for this reaction, and one inorganic phosphate is released. One phosphate group is attached to the carbamoyl group to form carbamoyl phosphate. So in mitochondria, ammonia reacts with bicarbonate. In cytosol, instead of ammonia, we have glutamine, which carries the amine group, and that amine group reacts with the bicarbonate to get the same product. In this case, the enzyme is carbamoyl phosphate synthetase 2. Okay, so we will focus on CPS1 for now. since CPS1 is the enzyme that is involved in the urea cycle. Okay. So the difference between CPS1 and CPS2 is only the amine source. So either the amine source could be ammonia or amine source could be glutamine. So you know glutamine carries the amine group, right? So it's just the nitrogen source is different. The substrate is different. The product is same. There are two isozymes, basically. These enzymes are two isozymes. Okay. So this is the first step in the urea cycle. So ammonia is converted into urea with the help of the enzyme carbamoyl phosphate synthetase. And this is the complete urea cycle where first carbamoyl phosphate is forming and then it reacts with ornithine to form citrulline, and then from citrulline we get arginosuccinate, arginine, and then finally urea is released and ornithine is recycled, and that's how the cycle keeps on going. But the first step, that is the major step, where the ammonia is incorporated into the cycle, is via carbamoyl phosphate synthetase, and the product we get is carbamoyl phosphate. I hope that's clear to everybody. Moving on to the next question now. Ammonia is transported from peripheral tissues to the liver, mainly in the form of option A, urea, option B, glutamine, option C, alanine, and option D, ammonium ions. Ammonia is transported from peripheral tissues to the liver, mainly in the form of option A, urea, option B, glutamine, option C, alanine, and option D, ammonium ions. So the correct answer is option C, alanine. So ammonia is transported from the peripheral tissues to the liver in the form of alanine. Okay. So this is a glucose alanine cycle, or it is also called Cahill cycle. So where alanine plays a special role in transporting the amino group. So since ammonia cannot enter the liver directly, alanine acts as a carrier of ammonia and of the carbon skeleton of pyruvate from the muscles to the liver. So it is doing two jobs. It is carrying the carbon skeleton from the pyruvate. So you know that glucose is metabolized into pyruvate by glycolysis. We have studied that in carbohydrate metabolism. So that carbon skeleton of pyruvate and the ammonia that is released from glutamate. Both of these groups are carried by alanine. So basically, pyruvate will get converted into alanine with the help of the amine group that comes from glutamate, uh, and glutamate will get converted into alpha ketoglutarate. This alanine will go into the bloodstream and then finally into the liver. So from muscles, from the peripheral tissues, alanine will carry the carbon skeleton and the ammonia or the amine group, the nitrogen group, into the liver. Once it enters the liver, it can again get converted into pyruvate, and alpha ketoglutarate can get converted into glutamate. And this glutamate can now be uh, broken down in, to release the ammonia, and that ammonia can enter the urea cycle as we saw in the previous slide. Okay, and the pyruvate can, you know, we have already studied about gluconeogenesis. Where does gluconeogenesis occur? Gluconeogenesis occurs in the liver, right? And pyruvate can get converted into glucose as and when required by the process of gluconeogenesis, and glucose can be released in blood in the form of blood glucose, when blood glucose level is low. Okay, so you know this part. So the carbohydrate metabolism part we have already discussed, right? So when blood glucose level is high, the glucose is broken down into pyruvate, and this pyruvate, along with the amine group of glutamate, becomes alanine, and glutamate gets converted into alpha ketoglutarate. This alpha ketoglutarate can enter the TCA cycle, and this alanine can come into the blood and then enter the liver again, where alanine will be broken down into pyruvate and alpha ketoglutarate will be broken down into glutamate. In liver, the glutamate can be converted into ammonia, and then ammonia can enter the urea cycle by the enzyme carbamoyl phosphate synthetase. And this pyruvate, when blood glucose level will go down or will be low, this pyruvate can again be converted back to glucose by gluconeogenesis in the liver, and this glucose can get released in the blood. Okay, so this is how glucose and alanine are interchanged between the peripheral tissue, between the muscle and the liver, through the bloodstream, and it carries, alanine carries the carbon skeleton and the ammonia of the pyruvate and carbon skeleton of the pyruvate and ammonia group from muscles to liver. The ammonia is excreted, and the pyruvate is used to produce glucose, which is returned to the muscles. Okay, I hope that's clear to everybody. Moving on to the next question. Which amino acid does not undergo transamination? Option A, alanine. Option B, glutamate. Option C, lysine. And option D, aspartate. Which amino acid does not undergo transamination? Option A, alanine. Option B, glutamate. Option C, lysine. And option D, aspartate. Which amino acid does not undergo transamination? Alanine, glutamate, lysine, and aspartate. The correct answer to this question is option C. So lysine and threonine, these are two enzymes that are, these are two amino acids that are that do not participate in transamination. So do you remember what was transamination? Transamination was the transfer of the amino group from one amino acid to an alpha keto acid to make another amino acid, to make glutamate basically. Okay, so we had amino acid that was reacting with alpha keto acid to make glutamate and another alpha keto acid. Okay. So all the amino acid except lysine and threonine, all the other amino acids, they participate in this transamination, and this is catalyzed by the enzyme transaminase. Later this glutamate will undergo deamination, and we'll get alpha ketoglutarate, which is an alpha keto acid, and the amine group will be removed in the form of ammonium ion or ammonia, and this ammonia can then enter the urea cycle, as we have already studied. So we have transamination and deamination occurring in the cells. So amino acid is transferring its amine group to the alpha keto acid to alpha ketoglutarate to become glutamate. This process is transamination, and lysine and threonine do not take uh, part in this transamination process. We'll study about lysine metabolism and threonine metabolism in the next sessions, where we'll see how lysine and threonine are transferring their amine, how they are getting metabolized. Okay, I hope that's clear. Moving on to the next question now. Elevated serum AST or ALT suggests damage to which organs? Elevated serum AST or ALT suggests damage to which organs? Option A, kidney and heart. Option B, liver and heart. Option C, lungs and kidney. Option D, pancreas and stomach. Elevated levels of serum AST or ALT suggest damage to which organs? Option A, kidney and heart. Option B, liver and heart. Option C, lungs and kidney. And option D, pancreas and stomach. Elevated serum AST or ALT suggests damage to which organs? Kidney and heart, liver and heart, lungs and kidney, pancreas and stomach. The correct answer is option B, liver and heart. So liver and heart gets damaged when there are elevated levels of AST or ALT. What are AST and ALT? So these are enzymes. ALT is alanine aminotransferase and AST is aspartate aminotransferase. They are also called SGPT and SGOT. So if you have seen some blood test reports, you must have seen the SGPT, SGOT levels mentioned. And generally, these are associated with liver and heart diseases. Okay, so AST, increased level of AST is associated with myocardial infarction, and increased level of ALT is associated with liver diseases. Okay, so mostly when liver is not functioning properly, the ALT levels increases, and when there is a defect in heart, when AST level increase. So increased AST level indicates myocardial infarction. So these are aminotransferase, or these are basically doing the transamination reaction. So we have already seen this example: aspartate becoming oxaloacetate and alpha ketoglutarate becoming glutamate. Similarly, this reaction takes place where alanine gets converted into pyruvate and sorry, alanine gets converted into pyruvate, and alpha ketoglutarate gets converted into glutamate. So alanine will transfer its amine group to alpha ketoglutarate, and alanine will become pyruvate, and alpha ketoglutarate will become glutamate. So aspartate aminotransferase and alanine aminotransferase, both AST and ALT, they are induced by glucocorticoids glucocorticoids, which favor gluconeogenesis, and elevated level of AST indicates myocardial infarction, and ALT indicates liver diseases. Okay. So I told you these are called SGPT and SGOT. So SGPT basically is the abbreviation of serum glutamate pyruvate trans uh, transferase. Okay, and SGOT serum glutamate oxaloacetate transferase. Okay, since the products are pyruvate and oxaloacetate, it is also named on the basis of that. I hope that's clear to everybody. So elevated level of ALT or AST in the blood indicates damage to liver and heart. Moving on to the next question. Which process frees ammonia for excretion? Which process frees ammonia for excretion? Option A, transamination. Option B, dehydration. Option C, deamination. And option D, glycolysis. Which process frees ammonia for excretion? Option A, transamination. Option B, dehydration. Option C, deamination. And option D, glycolysis. Which process frees ammonia for excretion? Transamination, dehydration, deamination, glycolysis. The correct answer is option C, deamination. So till now we have talked about transamination, where the amine group is transferred from one amino acid to alpha ketoglutarate to become glutamate. Then finally, this glutamate enters a process called deamination, where the amine group is released from the glutamate and it is converted back to alpha ketoglutarate. And the amine group is released in the form of ammonia. This ammonia can then enter the urea cycle, which we saw in the first slide, previous slide. So you have any amino acid, this will be the amino group of this amino acid is transferred to a keto acid, basically alpha ketoglutarate, and alpha ketoglutarate will take up the amine group to form glutamate, and corresponding keto acid will be released from this particular amino acid. Like in case of aspartate, we got oxaloacetate. In case of alanine, we'll get pyruvate. Okay. And alpha ketoglutarate will get converted into glutamate. This will happen in all tissues. This process is called transamination. Once glutamate is formed, glutamate is transported from the tissues to liver, and then, so I've told you how glutamate is transported to from the tissues to liver. with the help of alanine. Okay, and then this glutamate in the liver will get uh, oxidized into alpha ketoglutarate. So this amine group will be released in the form of ammonia. Okay, this is ammonia. Okay, so amine group will be released from the form of ammonia and we'll get a aldehyde group, which is a alpha keto acid, alpha ketoglutarate. And the enzyme that catalyzes this deamination is called glutamate dehydrogenase. Now, if you remember from the previous sessions also, I have told you if a dehydrogenase is involved, that means that it requires a reducing equivalent. Here we have NAD plus. NAD plus will get converted into NADH. That is, NAD plus is reduced to NADH, and in turn, glutamate is oxidized into alpha ketoglutarate. Okay, and ammonia is released. This ammonia can then get combined with bicarbonate with the help of the enzyme carbamoyl phosphate synthetase to form carbamoyl phosphate, and that carbamoyl phosphate can then enter the urea cycle for the excretion of ammonia in the form of urea from the body. So urea will be excreted out in the urine, and this is how ammonia is released from our body. Okay. So all the amino acids except which two amino acids? You remember what there were two amino acids that don't undergo transamination, lysine and threonine. So except lysine and threonine, all the other amino acids will get converted into their corresponding keto acids. And this reaction, this transamination can happen in all the tissues. And in turn, alpha ketoglutarate will get converted into glutamate. All the glutamate from all the tissues will be transferred to liver via alanine, via alanine pathway that we have already studied. That alanine will then again get converted into glutamate in the liver. In the liver, this enzyme glutamate dehydrogenase is present, which can convert the glutamate back to alpha ketoglutarate and release the ammonia group from the glutamate. So this is oxidative deamination of liver. I told you the oxidation is happening. Glutamate is oxidation. The glutamate is oxidized into alpha ketoglutarate. So oxidative deamination is happening. Deamination is happening because ammonia group is removed. Amine group is removed from glutamate in the form of ammonia. So deamination, and it's a oxidation. It's a oxidation reaction. NAD is reduced to NADH and in turn oxidizing glutamate into alpha ketoglutarate. That's why it's called oxidative deamination. Okay, and this process is occurring in liver. Why this is occurring in liver? Because glutamate dehydrogenase is present in the liver. Okay, and we get alpha ketoglutarate and ammonia is released. This ammonia will then be converted into urea, and urea will be excreted out in the urine. I hope that's clear to everybody. Moving on to the next question. The first step in the urea cycle involves the condensation of which two compounds? The first step in the urea cycle involves the condensation of which two compounds? Option A, ammonia and aspartate. Option B, ammonia and carbamoyl phosphate. Option C, carbamoyl phosphate and ornithine. Option D, ornithine and citrulline. The first step of urea cycle involves the condensation of which two compounds? Option A, ammonia and aspartate. Option B, ammonia and carbamoyl phosphate. Option C, carbamoyl phosphate and ornithine. And option D, ornithine and citrulline. The correct answer is option C, carbamoyl phosphate and ornithine. So I've already shown you this urea cycle. So we have discussed the first step where the ammonia is entering the urea cycle. How it is entering? Ammonia is entering by the help of the enzyme carbamoyl phosphate synthetase, where ammonia combines with the bicarbonate or the carbon dioxide to form carbamoyl phosphate with the help of hydrolysis of 2 ATPs. This carbamoyl phosphate will then enter the urea cycle. Uh, and the first step in the urea cycle is condensation of ornithine and carbamoyl phosphate to form citrulline. So ornithine along with carbamoyl phosphate will form citrulline, and the enzyme that catalyzes this reaction is called ornithine transcarbamylase. So ornithine transcarbamylase will condense carbamoyl phosphate and ornithine to form citrulline. This citrulline will then get converted into argininosuccinate with the help of enzyme argininosuccinate synthetase. Again, a synthetase is there. That means a ATP hydrolysis is there. And in the formation of argininosuccinate from citrulline, aspartate enters the cycle. So citrulline and aspartate with the help of the enzyme argininosuccinate synthetase will form argininosuccinate. This argininosuccinate will get converted into arginine by the enzyme argininosuccinate lyase, where fumarate will get released from argininosuccinate and arginine uh, will be the product, and arginine will then again get converted back to ornithine, releasing a molecule of urea. So that's how urea is released from the cycle and ornithine is recycled to react with next molecule of carbamoyl phosphate and citrulline will form. Citrulline will then cycle back to arginine, where arginine will cycle back to ornithine, releasing one more molecule of urea, and that's how urea is released, and this urea can be excreted out in the urine. Okay. And the fumarate that was released from argininosuccinate along with arginine, this fumarate will get cycled back to aspartate via malate and oxaloacetate. So if you remember, this is a part of TCA cycle. So fumarate will form malate by the enzyme fumarase, then malate will form oxaloacetate by malate dehydrogenase. This oxaloacetate can get converted into aspartate by aspartate aminotransferase. And then this aspartate can again enter the urea cycle. So this is how urea cycle is replenished. So we need aspartate and we need ornithine uh, for the urea cycle to cycle back. So basically, this is how all the cycles are interlinked and all the cycles regulate each other. So ammonia enters the urea cycle in the form of carbamoyl phosphate. How do we get carbamoyl phosphate? By the enzyme carbamoyl phosphate synthetase. Ammonia along with bicarbonate forms carbamoyl phosphate. This carbamoyl phosphate will enter the urea cycle, where first step of condensation will happen, where carbamoyl phosphate will condense with ornithine to form citrulline. The enzyme involved in this process is called ornithine transcarbamylase. Then citrulline along with aspartate will form arginosuccinate with the help of the enzyme arginosuccinate synthetase, which will require ATP since it's a synthetase, and this time ATP is hydrolyzed into AMP instead of ADP. So two high energy phosphate bonds are hydrolyzed in this reaction. Two pyrophosphates are uh, two inorganic phosphates, or a pyrophosphate is released. Okay. Then argininosuccinate will break down into arginine and fumarate. Fumarate can cycle back to aspartate to make more and more argininosuccinate, and arginine can cycle back to ornithine, releasing a one molecule of urea. Okay, that's how urea is released and urea cycle keeps on going. Okay, I hope this is clear. Moving on to the next question. Which organ is primary site for ammonia detoxification? Which organ is primary site for ammonia detoxification? Which organ is primary site for ammonia detoxification? Option A, kidney. Option B, heart. Option C, liver. And option D, brain. Which organ is the primary site for ammonia detoxification? Option A, kidney. Option B, heart. Option C, liver. And option D, brain. The correct answer is option C, liver. So you remember we have discussed that glutamate is transferred from all the tissues to the liver in the form of alanine, and then again gets converted back to glutamate, where glutamate is deaminated. Deamination reaction occurs by the enzyme glutamate dehydrogenase to release the ammonia. So primary site for ammonia detoxification is liver. Okay, so in liver, the uh, ammonia is released from the glutamate, and then the ammonia will enter the urea cycle in the form of carbamoyl phosphate. That's clear to everybody. Moving on to the next question. What is the major non-toxic transport form of ammonia in blood? What is the major non-toxic transport form of ammonia in blood? What is the major non-toxic transport form of ammonia in blood? Option A, glutamine. Option B, urea. Option C, free ammonia. And option D, pyruvate. What is the major non-toxic transport form of ammonia in blood? Option A, glutamine. Option B, urea. Option C, free ammonia. And option D, pyruvate. The correct answer is option A, glutamine. So glutamine is the major non-toxic transport form of ammonia in blood. So glutamine is the major non-toxic transport form of ammonia in blood. Glutamine is the amide of glutamic acid, and it provides a non-toxic storage and transport form of ammonia. So glutamate is converted into glutamine by so if you see, glutamine is the amide form of glutamic acid. So one amine group is added to the acidic group in the side chain of glutamate. So this reaction is catalyzed by the enzyme glutamine synthetase. Okay, again, a synthetase is involved. That means 1 ATP hydrolysis is done, and glutamine synthetase will then glutamine synthetase will then add one amine group to the glutamate to form glutamine, and this glutamine is the non-toxic form of ammonia carrier. It carries these amine groups, which can later be transported into the liver and converted back to glutamate, where in the liver, glutamate dehydrogenase can act on it and release the amine group in the form of ammonia. Okay. So in brain, glutamine is the major mechanism for removal of ammonia, while in liver, it's urea formation. So in liver, we have studied it about urea formation, right? So in brain, glutamine is directly used to remove the ammonia, and then in liver, ammonia gets converted into urea and then it is excreted out in urine. Circulating glutamine is removed by kidney, liver, and intestine, where it is deaminated by glutaminase. So, glutaminase will deaminate the glutamine to form glutamate, and then glutamate will be deaminated by glutamate dehydrogenase in the liver to uh, form alpha ketoglutarate and ammonia, which we have already studied during deamination. Okay, so glutamate will get converted into alpha ketoglutarate and ammonia in the liver. Glutamine is found in plasma at concentrations higher than other amino acids, which basically uh, basically it's a carrier of amine group, and it can transport the amine group to the liver via the bloodstream, and in the liver it can get converted into glutamate again by the enzyme glutaminase. Okay, so this is clear. So glutamine is the non-toxic storage and transport form of ammonia. Glutamine forms from glutamate by glutamine synthetase. Glutamine synthetase will use one ATP uh, to get the energy, and it will transfer the amine group to the carboxyl side, carboxyl group of the side chain of glutamate to form glutamine, and then glutamine will be transported to liver, and in the liver glutaminase will convert the glutamine back to glutamate, and this glutamate can be deaminated to form ammonia. I hope that's clear to everybody. Moving on to the next question. Which of the following enzymes catalyzes the conversion of arginine to ornithine in the urea cycle? Which of the following enzyme catalyzes the conversion of arginine to ornithine in the urea cycle? Option A, ornithine transcarbamylase. Option B, carbamoyl phosphate synthetase 1, CPS1. Option C, argininosuccinate synthetase. And option D, arginase. Which of the following enzyme catalyzes the conversion of arginine to ornithine in the urea cycle? Option A, ornithine transcarbamylase. Option B, carbamoyl phosphate synthetase 1, that is CPS1. Option C, argininosuccinate synthetase. And option D, arginase. Which of the following catalyzes the conversion of arginine to ornithine in the urea cycle? Ornithine transcarbamylase, carbamoyl phosphate synthetase 1, argininosuccinate synthetase, and arginase. The correct answer is option D, arginase. So we have seen in the urea cycle already the arginine that was forming from argininosuccinate. That arginine was getting converted back to ornithine, and one urea was released from arginine to ornithine. One urea was released, and ornithine was cycled back to form citrulline and then back to arginine. So we had citrulline, we had argininosuccinate, and then we were getting, so this was the urea cycle, right? This is a very brief version of urea cycle. So arginine was getting converted back to ornithine, where one urea was released out of the uh, where one urea was released out and arginine was cycled back to ornithine. And the enzyme that was catalyzing this reaction is arginase. Okay. So arginase catalyzes the reaction of arginine in converting arginine into ornithine. And in the process, one urea is released. And that's how urea cycle goes on. Ornithine will again condense with carbamoyl phosphate to form citrulline. Citrulline will form arginosuccinate, and arginosuccinate will again form arginine, which will again go into ornithine, releasing more and more urea. And this urea can then be excreted in the urine. Okay, so that's how urea cycle goes on. I hope that's clear. Moving on to the next question. Deficiencies in the enzymes involved in the urea cycle can lead to option A, hyperammonemia, option B, hypoglycemia, option C, hyperglycemia, and option D, hyperlipidemia. Deficiencies in the enzymes involved in urea cycle can lead to hyperammonemia, hypoglycemia, hyperglycemia, and hyperlipidemia. So the correct answer is option A, hyperammonemia. So as you know, urea cycle occurs in the body for clearing the ammonia in the circulation. So ammonia from the liver will be converted into urea so that it can be excreted out in the form in the urine, right? So if there are deficiencies in the enzyme that are involved in urea cycle, then the ammonia will not be able to get converted into urea, and then that ammonia will get accumulated, and we will have a condition called hyperammonemia, where the ammonia level will be high in the circulation. Okay. So deficiencies in the enzymes involved in urea cycle can lead to hyperammonemia. Moving on to the next question. Why must ammonia be rapidly converted in the body? Why do we need to clear out ammonia in the form of urine? Option A, it is poorly soluble. Option B, it is toxic, especially to central nervous system. Option C, it is required for energy. And option D, it aids digestion. Why must ammonia be rapidly converted in the body? Option A, it is poorly soluble. Option B, it is toxic. Option C, it is required for energy. And option D, it aids digestion. So the correct answer is option B. It is toxic, especially to the central nervous system. So that's why ammonia needs to be cleared out. Otherwise, we'll get a condition called hyperammonemia. The capacity of hepatic urea cycle exceeds the normal rates of ammonia generation, and the levels of serum ammonia are normally low. That it is very low, 5 to 50 micromole per liter. Okay. However, when liver function is compromised due to either some genetic defects or some liver disease, blood level can rise up to 1000 micromole per liter, and such hyperammonemia is a medical emergency because ammonia has direct neurotoxic effects on central nervous system. For example, elevated concentrations of ammonia in the blood causes symptoms of ammonia intoxication, which includes tremor, slurring of speech, somnolence, vomiting, cerebral edema, and blurring of vision. At high concentration, ammonia can cause coma and death. The two major types of hyperammonemia is acquired hyperammonemia and hereditary hyperammonemia. So, what are the toxic effects of ammonia on central nervous system? So, ammonia reacts with alpha ketoglutarate to form glutamate. So if ammonia is not cleared out from the system in the form of urea, it can react with the alpha ketoglutarate, because if you remember, in the liver, glutamate was getting converted into glutamate was getting converted into alpha ketoglutarate by releasing ammonia, right? Glutamate was getting converted into alpha ketoglutarate by the enzyme glutamate dehydrogenase, and ammonia was released, and this ammonia was entering the urea cycle. So if a urea cycle is defective, ammonia will stay here, and then ammonia can again get converted back to glutamate because this enzyme is reversible. Okay, and if it gets converted back to glutamate, there will be depleted levels of alpha ketoglutarate. And alpha ketoglutarate, it's a intermediate in the TCA cycle, right? Alpha ketoglutarate has to enter the TCA cycle. So if more and more alpha ketoglutarate is used up by ammonia to form glutamate, there will be depleted levels of alpha ketoglutarate that will lead to impaired function of the TCA cycle in the neurons, and that will lead to depletion of energy, because TCA cycle will ultimately lead to ATP production, and then there will be depletion of energy in the neurons. So this is one of the reasons why ammonia is toxic for the central nervous system, because ammonia, if not if ammonia is not getting converted into urea, ammonia can get converted into glutamate again, using up all the alpha ketoglutarate that is required for the TCA cycle, and then there will be impaired function of the TCA cycle. And TCA cycle, if TCA cycle is not functioning properly, then there will be depletion of energy in the neurons. Another reason is glutamate is used for glutamine formation. We have studied in the brain what was happening, and in other tissues also. Glutamate was getting converted into glutamine by the addition of amine group to glutamate. Glutamine is basically the amide form of glutamine, and amine group was added to the glutamate by the enzyme glutamine synthetase, right? Glutamine synthetase. And this glutamine synthetase is converting glutamate into glutamine. Now, if ammonia is present in a high level, ammonia is present in a higher level, what will happen? Ammonia will get converted into glutamate. Now, this glutamate will get converted into glutamine, and depletion of glutamate will happen. And glutamate is also required for the synthesis of a very important neurotransmitter, that is gamma aminobutyric acid. So, GABA is a neurotransmitter, and since glutamate will be used up for the formation of glutamine, there will be no glutamate present, and then there will be depletion of the neurotransmitter GABA, gamma aminobutyric acid, and this will again lead to toxic effects on the central nervous system. Excess glutamine comes out of the neural cells in exchange of amino acid tryptophan, and excess tryptophan leads to production of serotonin, which is an excitatory neurotransmitter. Okay, so now the third effect that is happening. So first effect was depletion of alpha ketoglutarate, so TCA cycle was not happening, so depletion of energy in the neurons was happening. Second effect was more and more glutamate formation was happening, that was leading to more and more glutamine formation, and glutamate was getting depleted, which was required for GABA neurotransmitter synthesis, and then depletion of this neurotransmitter led to toxic effects. Then more and more glutamine formation is also problematic, because more and more glutamine will come out of the neural cell and in exchange of tryptophan, and this tryptophan can lead to production of serotonin, which is an excitatory neurotransmitter. Then the fourth effect is astrocytes contain most of the total brain glutamine synthetase. Okay, so glutamine synthetase, I've told you, converts glutamate into glutamine, while neuron contains virtually all brain glutaminase. And glutaminase, what it does, it converts glutamate back to, glutamine back to glutamate. So glutaminase will convert glutamine to glutamate, and glutamine synthase will convert glutamate to glutamine. Okay. So in astrocytes, glutamine synthetase is present, and neurons contain glutaminase. Now, excess glutamine leads to osmotic imbalance in the astrocytes, which leads to water intake, and it swells up the astrocytes, leading to cerebral edema. Okay. Raised intracranial tension can lead to coma. And the fifth effect is ammonia-induced activation of NMDA and methyl D-aspartate receptors, increase nitric oxide formation, which in turn reduces the activity of glutamine synthase, thus reducing the elimination of ammonia in the brain. So basically, higher levels of ammonia or defect in the urea cycle can actually lead to several different toxic effects in the central nervous system. First is depletion of alpha ketoglutarate, leading to loss in TCA cycle and leading to depletion of energy in the neurons. Second is glutamate formation can form glutamine, thus depleting the levels of glutamate. Low levels of glutamate can lead to low level of gamma aminobutyric acid, which is an important neurotransmitter. Higher levels of glutamine can actually come out of the neural cells in exchange of tryptophan, and it can lead to production of serotonin, which is an excitatory neurotransmitter, which can lead to all the seizures that are observed during hyperammonemia. Then astrocytes can swell up due to the osmotic imbalance uh, because of the excess glutamine production. And then final effect is ammonia-induced activation of NMDA, which can increase the NMDA receptors, which can increase the nitric oxide formation, which can reduce the glutamine synthetase activity, thus reducing the elimination of ammonia in the brain. Okay. I hope that's clear. Moving on to the next question. Which enzyme deficiency is the most common urea cycle disorder? Option A, arginase. Option B, carbamoyl phosphate synthase 1. Option C, ornithine transcarbamylase. And option D, NAGS or N-acetyl glucosamine. Which an enzyme deficiency is most common urea cycle disorder? Option A, arginase. Option B, carbamoyl phosphate synthase 1. Option C, ornithine transcarbamylase. And option D, NAG. So the correct answer is option C, ornithine transcarbamylase. So ornithine transcarbamylase, if you remember, it does the condensation reaction between carbamoyl phosphate and ornithine. This is the first reaction in the urea cycle where ornithine and carbamoyl phosphate condenses to form citrulline. The citrulline then forms argininosuccinate, and then argininosuccinate forms arginine, which can cycle back to ornithine, releasing one molecule of urea. So this is a part of urea cycle, and this is the first reaction of urea cycle where the ammonia in the form of carbamoyl phosphate enters the urea cycle by condensing with ornithine to form citrulline. So ornithine carbamylase catalyzes this reaction, and deficiency of this enzyme is characterized by HHH syndrome. So it stands for hyperornithinemia. Obviously, if this enzyme is not present, then ornithine will keep getting accumulated and we'll have hyperornithinemia. Hyperammonemia, because ultimately ammonia is getting converted to urea. So if this enzyme is not functioning properly, or if this enzyme is deficient, ammonia will get accumulated and we'll have a condition of hyperammonemia, and we'll have another condition called citrullinuria. As ornithine is not available in the mitochondria, lysine is carbamylated to form homocitrulline. So lysine will get converted into homocitrulline and we'll have citrullinuria, because citrulline will get excreted out in the urine. Okay. So ornithine and ammonia will get accumulated and homocitrulline will be excreted out of the urine, and we'll have the HHS syndrome. So this is a characteristic of ornithine transcarbamylase deficiency. I hope that's clear. Moving on to the next question. Deficiency of which of the following enzyme causes friable tufted hair? Option A, arginase. Option B, carbamoyl phosphate synthetase 1. Option C, ornithine transcarbamylase. And option D, argininosuccinate lyase. Deficiency of which of the following enzymes causes friable tufted hair? Arginase, carbamoyl phosphate synthetase 1, ornithine transcarbamylase, and argininosuccinate lyase. The correct answer is option D, argininosuccinate lyase. So this is another part of urea cycle. So in urea cycle, ornithine was condensing with carbamoyl phosphate to form citrulline. Citrulline was getting converted into argininosuccinate, and then argininosuccinate breaks down into arginine and fumarate. Fumarate gets cycled back to argininosuccinate in the with the intermediate of aspartate, and arginine makes ornithine and releases a molecule of urea. So this is a part of urea cycle. Okay. And the enzyme argininosuccinate lyase basically breaks the argininosuccinate into arginine and fumarate. Deficiency of this uh, enzyme leads to friable tufted hair or trichorexis nodosa. So this is a characteristic feature of this enzyme deficiency. This is a TEM image of the hair. So the hair becomes very brittle and it can break easily. I hope that's clear. Moving on to the next question. Which of the following molecules allosterically activates carbamoyl phosphate synthetase 1, the enzyme responsible for first step of urea cycle? Option A, N-acetyl glutamate. Option B, ornithine. Option C, citrulline. And option D, arginine. Which of the following molecules allosterically activates carbamoyl phosphate synthetase 1, CPS1, the enzyme responsible for the first step of urea cycle? So the correct answer is option A, N-acetyl glutamate or NAG. So I told you in the one of the previous slides, we have two isozymes of carbamoyl phosphate synthetase, CPS1 and CPS2. Both of them catalyzes the same reaction. The only difference is CPS1 catalyzes the reaction with ammonia and bicarbonate to form carbamoyl phosphate, and CPS2. So this reaction is carried out by CPS1. CPS2 instead of ammonia uses glutamate as a substrate. So glutamate is the ammonia carrier, and CPS2 will catalyze this reaction. CPS2 catalyzes this reaction to make the same product. Okay, so the only difference is in the substrate. CPS1 uses ammonia directly and CPS2 uses glutamate. So the difference between CPS1 and CPS2, CPS1 acts in mitochondria and CPS2 acts in cytosol. And CPS1 is involved in the urea cycle, whereas CPS2 is actually involved in the pyrimidine. So this carbamoyl phosphate that we get is goes into the pyrimidine synthesis pathway. This we will study in the upcoming sessions. Then CPS1 has a positive effector, that is N-acetyl glutamate. So N-acetyl glutamate enhances the activity of CPS1. CPS2 does not have any positive effector. Sources of nitrogen, I told you, in CPS1 it is ammonia, in CPS2 it is glutamine. Inhibitor, there's no inhibitor for CPS1 known, but the CTP is an inhibitor of CPS2. Okay, so allosteric activation of CPS1 uh, is done by N-acetyl glutamate, NAG. Okay. Next question is, which of the following molecules can be used for treating the disorders occurring due to blocks in the first two steps of urea cycle? Which of the following molecules can be used for treating the disorders occurring due to blocks in the first two steps of urea cycle? Option A, arginine. Option B, benzoate. Option C, phenyl acetate. And option D, both B and C. That is benzoate and phenyl acetate. So the correct answer is option D, both B and C, that is benzoate and phenyl acetate. So benzoate and phenyl acetate can actually be used as a treatment strategy in case the first two enzymes of the urea cycle are deficient. So benzoate will be converted to benzoyl coenzyme A, which will then get converted into hippuric acid or hippurate. This can directly get excreted into urine because it's a soluble compound. So benzoate will get converted into hippuric acid, and similarly, phenyl acetate can get converted into phenyl acetyl glutamine, which can also get excreted into urine. So phenyl acetate and phenyl acetate of phenyl butyrate and benzoate can be administered in the diet if the first two enzymes of the urea cycle are deficient. They can combine with glycine and glutamine respectively, and they can form hippuric acid and phenyl acetyl glutamine. And these products, since they are soluble, they can be excreted in the urine. Is that clear to everyone? So this is the last question of this session. If there's any doubt, you can post in the chat box. Okay, so with this, we can end the session now. Thank you all for joining. I'll close the session.